Good morning, everyone. Our lesson for today is about the microbial cell wall. This lesson focuses on the bacterial cell wall. Specifically, it will make you understand how the cell wall functions, what is its composition, what are the basic types of the cell wall, and how it relates to microbial classification. At the end of this lesson, you are expected to be able to describe the functions of the cell wall, identify the composition of the cell wall, differentiate gram-positive from gram-negative cell wall, and relate the gram-stain mechanism to the structure of the cell wall. Before we begin our discussion, I will be asking you this question. Does the composition of the cell wall determine its functions? I hope you will be able to answer that one at the end of this lesson. So the cell wall of the bacterial cell is a complex, semi-rigid structure responsible for the shape of the cells. So if you can see here, this is the electron micrograph of a representative bacterial cell, and being pointed here, this arrow is the cell wall, no, of these bacteria. So the cell wall surrounds the underlying fragile plasma or cytoplasmic membrane and protects it from the and protects it in the interior of the cell from adverse changes in the outside environment. And almost all prokaryotes have cell walls, so the bacteria as well as the archaea, they have their cell walls. And even some groups of eukaryotic organisms now have cell wall, like the fungi. So what are the functions now of the cell wall? So the cell wall prevent bacterial cells from rupturing when the water pressure inside the cell is greater than that of the outside the cell. And it helps maintain the shape of a bacterium and serves as a point of anchorage for the flagella. So as the volume of a bacterial cell increases, its plasma membrane and cell wall is extend as needed. Clinically, the cell wall is important because it contributes to the ability of some species to cause disease, and it is the site of action, no, of some of the antibiotics. So at the end, no, or the last module, we'll talk about um, uh, control of growth of microorganisms, and we will be talking about actions now of some antibiotics. So the chemical composition also of the cell wall is used to differentiate major types of bacteria, which we will talk about later on. So what is the composition of the cell wall? What are those that comprises the cell wall? So the bacterial cell wall is composed of a macromolecular network called peptidoglycan, which is also known as myelin. This is present either alone or in combination with other substances. So the peptidoglycans consist of a repeating disaccharide attached by polypeptides to form a lattice, no, like a net, that surrounds and protects the entire cell. So the disaccharide portion is made up of monosaccharides called the NOG and the NAM. So NOG stands for N-acetyl-glucosamine, while NAM stands for N-acetylmuramic acid. Okay, so these are actually glucose derivatives. So this is the chemical structure of both of these disaccharide. So disaccharide because this is a monosaccharide and another monosaccharide. So they are joined together by this glycosidic bond. So it is actually a disaccharide, but it is a glucose derivative. Okay. So together, the carbohydrate backbone, so the NAG and the NAM, forms the glycan portion. However, no, connecting this glycan portion, the repeating disaccharide, are the tetrapeptide side chains. So this makes up the peptide portion. So they make up the peptidoglycan. So that means a combination of a peptide, a tetrapeptide, and a glycan portion. So we already know the glycan portion that is made up of disaccharide, the NUC, and the NOM. But how about the tetrapeptide side chains? So it is a tetrapeptide side chains, meaning there are four peptide bonds found. However, there are only four amino acids present because the last peptide bond is formed between the L-alanine and this functional group of the N-acetylmuramic acid. So together with alanine, commonly found in some or in most bacterial cell wall are glutamic acid, diaminopimelic acid, and D-alanine. Although the composition may differ in some species of bacteria. Now, there are two types of cell walls in bacteria. So, a cell wall could be gram-positive or they could be gram-negative. How do they differ from each other? A gram-positive cell wall, okay, has a thick layer of peptidoglycan, okay, many layers. So, they form a thick and a rigid structure. Also, gram-positive cell wall contain teichoic acids. So, two types of teichoic acids are found in gram-positive cell wall, the lipoteichoic acid and the wall teichoic acid. Wall teichoic acids are found embedded within the peptidoglycan layer, while lipoteichoic acids are those that span from the peptidoglycan layer and is embedded towards the cell membrane of the gram-positive bacteria. The gram-negative cell wall, on the other hand, contains only a thin layer of peptidoglycan. No, but what makes it different from gram-positive, aside from having a thin layer of peptidoglycan, is the presence of the outer membrane. So if you notice here, aside from the inner membrane or the plasma membrane of the gram-negative bacteria or bacterial cell, it also has an outer membrane. So the peptidoglycan is like a filling, no, between these sandwiches or between these lobes. So parang they make up a sandwich with a peptidoglycan filling. So actually the outer membrane together the peptidoglycan makes up the cell wall of this gram-negative bacterial cell. Now the gram-negative bacterial cell wall has outer membrane with LPS that are embedded in the phospholipid bilayer. So the LPS or the lipopolysaccharide is composed of the lipid A, core polysaccharide, and O polysaccharide. So peptidoglycan here in gram-negative bacterial cell is bonded to lipoproteins. So this is the lipoprotein. So the lipids are covalently linked to proteins. So lipoprotein, a combination of a lipid and a protein, found in the outer membrane and is in the periplasm. The periplasm is the gel-like fluid between the outer and the inner membrane or the plasma membrane. Okay, so this gel-like fluid here, where the peptidoglycan layer is embedded, is the periplasm. But what connects the peptidoglycan to the outer membrane are structures called, or are molecules called lipoprotein. And you have here the lipopolysaccharide component. Now, the outer membrane of gram-negative cell wall, as we have said, is composed of lipopolysaccharides, lipoproteins, and phospholipids. And this outer membrane has specialized functions. The strong negative charge is an important factor in evading phagocytosis, being eaten by other cells, and the actions of complements. So complements are those that are released by cytotoxic T cell to kill those cells that are bounded with antibodies. So in order to evade that, the gram-negative cell wall has outer membrane that could help the cell survive, the action of complement. The outer membrane also provides a barrier to certain antibiotics like penicillin. So penicillin actually targets the peptidoglycan layer. So with a barrier, the penicillin could not really penetrate into the peptidoglycan layer of gram-negative bacterial cell. Aside from that, it could also be a barrier for digestive enzymes such as lysozyme, detergents, heavy metals, bile salts, and certain dyes. So as what we've said, lipopolysaccharides are found in the outer membrane. It is a large complex molecule that contains lipids and carbohydrates and consists of three components, the lipid A, core polysaccharide, and O polysaccharide. The lipid A is the lipid portion of lipopolysaccharide and is embedded in the top layer of the outer membrane. So when a gram-negative bacteria die, they will release lipid A, which functions as an endotoxin. Lipid A is responsible for the symptoms associated with infections by gram-negative bacteria such as fever, dilation of blood vessels, shock, and blood clotting. The core polysaccharide, on the other hand, is attached to lipid A and contains unusual sugars. The role is structural, so they provide stability for the outer membrane. Lastly, the O polysaccharide is the one that extends outward from the core polysaccharide and is composed of sugar molecules. So the O polysaccharide functions as an antigen, the one that elicits immune response in the cell, and is useful for distinguishing species of gram-negative bacteria. Now, these bacteria, because of the differences in bacterial cell wall, a technique was developed by Hans Christian Grams, to in order for us to determine which type of cell wall the certain bacteria has. It's very important because in early diagnosis, especially in medical cases when infection has already gone severe, so the first thing to do is to determine the gram or the gram reaction or the cell wall of the bacteria in order to administer the proper antibiotics, and also in classification, so gram staining is needed. So this is the steps on how you would determine the cell wall composition of bacteria, if the bacterial cell has gram negative or gram positive cell wall. So the mechanism is based on differences in the structure of the cell walls of gram positive and gram negative bacteria and how they react to the various reagents. So the reagents are crystal violet as the primary stain, mordant, the iodine as the mordant, then decolorizer, alcohol as the decolorizer, and saffronin as the counter stain. So what they do first is you will also be doing this in your laboratory actually. So what you'll do first is you stain the cells with crystal violet. So the crystal violet will bind directly to the peptidoglycan layer of the bacterial cell wall. Then you add an iodine as the mordant. So what iodine does is inside in the peptidoglycan layer, the iodine will bind with crystal violet, so they form a crystal structure. So that when you wash the stain, it will not be washed out because they are trapped inside the peptidoglycan layer because they had formed a crystal structure. Now, an alcohol is applied as a decolorizer. So the decolorizer, the alcohol will degrade the outer membrane of the gram-negative bacteria. Now, so if crystal violet and iodine has already formed a lattice around here and they cannot be washed out because of the thin layer of the peptidoglycan layer, and because of this, but when the decolorizer is applied, it will degrade the outer membrane and some of the peptidoglycan layer so that the crystal violet and the iodine is also washed out. While in gram-positive bacteria, it will not be washed out because there is a thick layer of peptidoglycan. So what happens is that during crystal violet staining, both gram-positive and gram-negative cell will still be colored violet or blue. After the application of mordant, both of the cells will be colored blue or violet. However, after decolorizer, the gram-negative bacterial cell will be colorless, while the gram-positive bacterial cell will remain blue. And last, after decolorizing, saffronin is added as a counter stain. Okay, so what happens if you add saffronin to a gram-negative cell? Of course, since a gram-negative cell has already colorless, it will form pink to red color. It will just retain the saffronin color, which is red. So it could be, depending on the application, pink to red color. However, since the gram-positive cell has still crystal violet in its cell wall, the application of a red color will make it a more violet darker. So it could be violet to blue. So after your gram staining reaction, you will notice if it is pink to red, that means the cell is gram negative. If it is blue to violet, then that bacterial cell is a gram positive bacteria. So here's a result of one of my thesis students who has investigated antibiotic resistant bacteria no in swine. And this is his comparison of a gram reaction. You have here an E. coli, which is a gram negative bacteria, and Staphylococcus vitellinus, which is a gram positive bacterial cell. So that ends our lesson. No? So in order for you to review this lesson, you should be able to answer these summary questions. Why is cell wall important in a microbial cell? What are the components of the microbial cell wall? How do gram-positive cell walls differ from gram-negative cell walls? And what is the role of gram staining in microbial identification? Now, before we end, I would like to thank Miss Maria Sherlita Esrsal for helping me in making the slide presentation. So if you have questions, you can contact me in my email, or you can contact your lecturer in their email addresses, or you can key in also your questions in the group chat in the Facebook Messenger. Okay, so thank you very much, and see you again in our next lesson.